I will be posting the recordings later on this afternoon. So you'll be able to find links to those in the presentation channel on Slack. And I think without further ado, I'm really happy to be able to introduce Dr. David Koelle from the University of Washington, who is an expert on immunology and developing vaccines and T cells and antibodies and just about everything, I think. David, take it
Okay, great.
away.
Yeah, thank you. Thank you, Sandy, and welcome everybody to the hackathon and all about antibodies. I'm going to share my screen, see if this can work here. And uh, let me make it big. Do you see the presentation?
Yes.
Okay, it says something like mind the rash, right? Okay, great, great. Um, and just my disclaimers are on the lower right, and I might mention a few products, or vendors or something like that, but it doesn't mean I necessarily endorse them. But we're in a dynamic commercial space here. So see if this can work here. Going to the next slide, and as Sandy mentioned, it does sort of take a village, takes a big group, and I want to point out Chris McClurkin, who's the second from the left, who graduated from Sandy's program about 25 years ago at the community college and has actually been with me for 25 years and was a physicist and nuclear Navy and veteran and so forth, and now is having a great time in our lab. And he actually did some of the data I'm going to show you. And then our single cell sequencing, which is what sort of enabled our work. Li Chen Jing on the far left is the main person behind that. So objectives, for today is sort of introduce you to this virus that we're working on. And you probably already know it intimately. It's probably in your body. And the motivation, why are we doing this? There are some unmet medical needs, but also some cool things we can maybe learn. And there is a lot of naked capitalism around antibody therapy these days. It's something like, I got a slide at the end, like $20-25 billion a year or something like that. So there's kind of a gold rush going on in biotech with antibodies. I'll mention some of the main technologies we use, starting with single cell sequencing to get antibody sequences, creating human monoclonal antibodies, which we actually order out like ordering pizza. Now, you can do it yourself in your lab, but we find it more cost effective to use a commercial vendor for that. And then how are we going to determine whether these candidate antibodies are what we want or not, and then to summarize with what we might have learned. So the pathogen that we're talking about is called varicella zoster virus, or VZV, otherwise known as the pathogen that causes chickenpox. And the lifestyle of this pathogen is sort of summarized in this diagram. On initial infection, people who don't have any immunity, it causes this body-wide infection called chickenpox. And if you were born before about 1995, you probably had chickenpox. You probably remember your siblings having it. You breathe in the virus, it spreads through the blood. And then your immune system deals with it. And it actually, see if you can see my pointer. Let me see if I can wake up my pointer.
We see it.
Do you see it? Okay, so here it's sort of showing in nerve cells and near the spinal cord. It can come back later in life to cause a rash in the distribution of one nerve. Typically, we call it zoster or shingles. And yeah, if you were born before 1995, you might recognize this on yourself or one of your siblings or something. And we do have some vaccines now that can prevent this chickenpox. They're live attenuated virus. People either get the 4-in-1 MMRV, or it can also a standalone vaccine. When it comes back as shingles and goes up when you get older, immune compromised people, or just sort of bad luck, causes this rash. And that, it's painful and the immune response eventually deals with it. People lose time from work, have to maybe take narcotics and stuff, which is a problem. Occasionally it reactivates in bad places, like into the brain or into the eye, which can be really severe, lead to vision loss and so forth. And we have a partially effective vaccine. It was licensed before, and now we have a relatively effective vaccine to prevent this recrudescence, this return, although it has some side effects, this vaccine. And this is just a little diagram of our friend here. It's an enveloped virus, and it has these proteins on, just sort of remind you maybe of SARS-CoV-2, cartoons we saw on the side of buses and everything. And it has a DNA genome in the middle. It's pretty big for a virus. It encodes 70 proteins. SARS-CoV-2 is about 15, influenza is 7. Not very much strain variability, which we like. If we're going to make an antibody, we don't want something where there's all sorts of variation in the envelope proteins, right? And we would tend to think that antibodies against the surface envelope proteins would tend to be the most preventative or most therapeutic, because if antibodies bind to the outside of the virus, they can prevent it from interacting with a receptor and getting into a new cell. So we know this, and it's been completely, sequenced. Sorry, when I click on things, the slide seems to be going forward a bit. And you're probably very, you can intuit this sort of idea behind an antibody therapy, that if here's the virus particle with these envelope glycoproteins on the outside, when it wants to get into a cell, it has to have a receptor ligand interaction with a receptor on the surface of a cell. And the green Y-shaped things, and we'll come back to more diagrams of these a little more detail a little later, if they bind to the outside of the surface envelopes, they can prevent the receptor binding. And they can also become attractive from the back end of the Y, from sort of the stem of the Y, if you will, to what's called an FC receptor, this purple little guy here, which can then promote a phagocyte, an eating cell, to sort of interact with that virus, antibody-coated virus particle, and ingest it and digest it and render it harmless. So probably already know this, but this is sort of some intuition behind how antibodies can help. And we do have antiviral antibody drugs. So, for example, for SARS-CoV-2, this is just one of them. It's not meant to be an endorsement. Once SARS-CoV-2 popped up in late 2019, early 2020, I would say that probably over 100,000 human monoclonal antibodies were created to the spike protein within pharma and academia. And they were sort of screened to get sort of the best. And as you know, it mutated, the spike protein mutated in different generations of monoclonal antibody drugs, had to come out. But for VZV, we really don't have a monoclonal antibody product. We have this so-called pooled immune globulin. So this is a product made from lots and lots of blood donors who have decent levels of mixed polyclonal antibodies in their blood. And they'll literally just take the whole plasma, separate out the antibody fraction, the IgG fraction, and then lyophilize it and kind of, freeze dry it down. And this is as good as we have for therapy, for treating people if our standard drugs aren't really working well, is this Verazig product. So one of the motivations we have is, can we make or find a next generation good monoclonal antibody? I mentioned that we also are doing this research because it's cool and we want to learn cool stuff. And so sort of a dual motivation. And the idea behind, what might be cool is having to do with where antibodies are made in the body, just how we have an immune response to an infection. And this is kind of the old school over here on the left. There are these lymphoid organs, the lymphocytes are all born in the bone marrow. And then some cases they get educated in the thymus, but for B cells we're talking about, not B cells that make antibodies. Traditional thinking is that they get primed, they get programmed in lymph nodes, then they end up living in the bone marrow and in the spleen where they can pump out antibodies. Antibodies are proteins with 4 different chains, typically molecular weights around 125,000. They diffuse around in the blood and they go to where the infection is. That's old school. They are soluble. There's barriers to their diffusion, but they can travel. New school is that actually B cells, or these little purple guys here, or antibody producing cells, may actually reside locally where the infection is and make high concentrations of local antibody. And this is a little bit, counter-dogmatic, a little bit new school, but we wanted to investigate whether this was true by so-called mining the rash, by actually getting samples of the human rash of shingles and seeing if we could find virus-specific B cells in the rash. So that's what we set out to do. So here is a rash of shingles, and this is a dermatologist skin biopsy tool. And if you've ever had a skin biopsy, it's not that big of a deal. This is about a 3 millimeter little diameter thing. Little Novocaine gets put in the skin. It's sharp on the end. You sort of twirl it around, and you end up with this punch of tissue where there's an epidermis and then dermis and then some fat and stuff below. And that's, we pay our subjects informed consent and so forth, and we get our samples. And then what we want to do is make a single cell suspension, because we want to do single cell sequencing and with a couple of different targets. And we're using a commercial solution here to do this. Is this is like a 50 mil conical bottom tube, sort of an upside down tube, and it's got this sort of cutting teeth on this special little propeller thingy. And all this yellow unit here is a little motor with a little magnetic induction drive. So there's a magnet inside the lid of the tube, and it with a sort of a no-touch thing, and it sort of spins this lid, if you will. And there's some digestive enzymes in there that break down connective tissue and collagen kind of between the cells. So after a while in this special tube, the tissue is kind of digested to sort of single cells. And of course, there's some, grist and some, connective tissue in there. So we put it through a filter to try to get to a single cell prep. And then we go into a commercial drop-seq solution. So some of you may, be able to intuit which company this is. I won't necessarily name it, but what it does is it combines barcoded beads. So single individual little beads. Those are shown here in the barcoded primer gel beads. And each one of these has reverse transcription chemistry in it. And then here come the cells coming down through this other track here. And there's a creation of an emulsion with micro droplets, nanoliter droplets of aqueous phase, each one of which contains one bead and one cell. Now, some are going to contain a bead but not a cell. Some are going to contain a cell but not a bead. Some are going to contain two cells. So this is a sensitive and expensive little step. This is about $1,500 a sample here to create the emulsion. But once you create the emulsion, then you put it in the PCR machine at a certain temperature and time, and all of the mRNA in that little microdroplet is converted to cDNA. And of course, now the cDNA is quite stable. So now you can go ahead and create a library. There's a further PCR amplification step. There is a UMI, a unique molecular identifier, included in the cDNA collection. So every mRNA molecule gets a unique tag, so you can actually count the mRNA molecules per cell. And then every gel bead has a unique tag too. So that's how you tell which cell is which. So once you go through the wet lab process and you sequence, so this is on the Illumina, and we use a core facility on Illumina, and we get these giant FASTQ files back. Then we're now switching from wet lab to dry lab. And those of you who are more into bioinformatics might be handed a file name or sort of access to a server with some giant FASTQ files. And then your job is to sort of demultiplex and decode this into finally what's called a gene barcode matrix. So for each cell, you have a giant flat file of the names of human genes and then the number of counts for that gene. So I'm going to go on from here. This is the well-known commercial vendor for this particular technology. And once you do that, so we've got our skin biopsies and these huge Illumina files. There's some QC. We're going to dump the low-quality cells, align all of the cDNA rates to the human genome, normalize and integrate the data, meaning that if we have separate runs, there's ways statistically of sort of combining the data so fair is fair. And then we can actually look up the cell type signatures by matching our data to previously published signatures. And from a skin biopsy, we might see something like this. So this is a UMAP. This is arbitrary space. There's no physical reality behind this, but each dot is a cell. And then we've labeled the main nests of cells by what they look like from their mRNA transcription profile. So B cells, for example, are going to have a lot of reads for IgG or for immunoglobulin, and that's how they get assigned to be B cells. We also in the lab work on T cells and so forth. These are skin cells, keratinocytes, these are dermal cells, and so forth, other cells that were in the sample. So our surprise, there were B cells in the skin, and we wanted to know, were there antibodies that they were making virus specific or not? So we found this nice nest of human B cells in the skin. There's a little, introduction. I know this is the main theme of your talk today, but just take a look at an IgG molecule. And in this schematic, so it's actually got 4 polypeptides, 2 copies of the heavy chain in blue, and 2 copies of the light chain in sort of a purple. And this is showing here out on the tips, this variable regions, the variable heavy and variable light chain. And here they are binding to an antigen. This might be a viral protein or something, this green blob. Is that we know that this repertoire is the term we use, variability, it's super variable within person, maybe around 10 to the 13th or something, different combinations of variable heavy and variable light chains, because there's a whole genetic mix and match, hyper recombination and gene editing mechanism in our bodies and lymph nodes that can generate this great variability. I'm not going to talk about that anymore here today, but this is that we're mining nature's recombination magic with this huge number of combinations that we can get either within a person or within a population. And looking at this in another cartoon, people are always looking at cartoons. Here's the same, basic thing, but now notice these little loops, these three little blue things and these three little, orange things here. Those are termed in the business the CDR's complementarity determining regions, and these actually are the business end of binding to the antigen, and these are the most hypervariable regions. And here's even another little close-up-y sort of cartoon thing. These blue upside down, U-shaped things. These are going to have very variable amino acid sequences. That's where the heart of the variability is. And really, if the CDR3 is the very most variable. And so between the CDR3 of the heavy chain and the CDR3 of the light chain, there's a huge number of binding options. And here's yet another picture, and this is based more on an actual space filling sort of or backbone diagram. Here's what it looks like in the book over on the left, but this is actually more of a real structure. And in purple here is one of the heavy chains, and in green is one of the light chains. And this little squiggly guy right here, and this little squiggly guy, and this little squiggly guy, and then these loops here. So this is a little bit closer to what it actually might look like for an actual structure. I'm not rotating it or anything here in terms of the actual structure. So here's a little look at some of our real data. So we submit all these things for sequences. We actually make a special library for antibody sequencing. Another name for antibody is BCR, B cell receptor. It's the same thing. So BCR-seq antibody sequencing. And just to give you a bird's eye view of what we're looking at here, here's sort of our patient ID number. And sisters means that in this biopsy, wow, from this person, we got 306 identical B cells. Some B cell clone in there was having a lot of babies and a lot of reproduction in the skin. So that's already sort of suspicious that it might be responding to an infection. And these are actually the amino acid sequences in the single letter code. And hopefully you all know your code of the heavy chain and the light chain. So this, of course, has been translated by the computer for us from actually a nucleic acid sequence. So sitting in the computer in some other rows that I'm not showing you, pardon me, some other columns I'm not showing you, are the nucleic acid sequences underlying the heavy and the light chain. So now I have some candidates, and I want to progress to the next step of saying, is my candidate actually going to bind to the virus or not? And so we're turning to synthetic biology and protein expression here to answer that question. So we prioritize through all these antibody sequences some pairs. The word pair is key in immunorepertoire sequencing. You got to have the alpha and the beta. You got to have the heavy and the light together in a pair to sort of start synthesizing immune receptors and testing them. And we'll assemble the DNA sequences, so more of a dry lab task where we'll take that line listing, that crude line listing, and sort of turn this into some full-length sequences, which, and then the back end that we use down here, this FC part, we're going to use sort of the generic, it's called IgG1. I'm not going to go into any of the details, but regardless of what antibody isotype it came from, we're going to express these as an IgG1. That refers to the FC part, to the back end. And I'm going to push the button and send some DNA sequences to a company where I've also got a purchase order there. So because they're going to charge me some money. And at the company, they're going to make some oligos that encode the heavy and the light chain. They're going to clone this into a plasmid and sequence confirm it, express it by transiently transfecting some cells with that plasmid. The immunoglobulin is going to be secreted. And at the company, they're going to purify it with protein G, which binds to the back end, and do some QC, and they're going to send me a package in the mail. And then I'm going to get happy when they send me a package in the mail, and also send me some plasmid if I want to make more myself. And let's see if we can go to the next slide. And this is just sort of a cartoon of the plasmid that they're going to make. So here's the heavy chain, this long guy here. There's the variable heavy. Here's the light chain. They use two different promoters. It's sort of a traditional plasmid. You can also do this yourself, but we find it cost effective to send the sequence to a company, have them do this, and then just send us the antibody. And here's an example of the QC report that we would get back for every antibody. So here's a nice SDS page, silver stain. And when we reduce, it breaks up those to the heavy and the light chain. So heavy is here, light is here. And a non-reducing gel, they're still stuck together by disulfide bonds. So there it is a single high molecular weight molecule. Here's an HPLC. It's one nice band. We get sort of our QC report. And so we're ready to rock now with this candidate monoclonal antibody from a commercial vendor. So are they any good? So we're going to screen them against whole virus. And the answer, the check mark means we've done it. We've got an answer. And the answer is yes. And I'll show you some sample data. Do the virus-specific B cells go to the rash? And then which protein do the positive human monoclonals bind to? Because I mentioned there were about 70 proteins encoded in this virus. And we've got some data on that. I'll show you one example. The medical side, which is, do these monoclonal antibodies actually block infectivity? That's still a work in progress. So I'm not going to show you anything on that. So how in the heck are we going to figure out whether it sees the whole virus? So we're going to use fluorescence microscopy, which is another common sort of workstation and skill in our lab. And we are using a version of the VZV virus that has GFP, or really enhanced GFP, eGFP inserted into it. So this is just kind of a cartoon of the VZV stick DNA. It's about 150,000 base pairs long. And as a fusion with this one protein, protein 66, some other researchers stuck in a copy of GFP as a fusion. So what you're seeing here is a monolayer of cells. You can't see the cells, but there is a nice flat layer of cells growing on a plastic dish here, and they've been infected with this GFP virus. And you can kind of see individual cells, and you can see how there's sort of little nests of them. That's what we call a plaque in the business. So a virus got into one of these cells, and it's been spreading from cell to cell inside that plaque. So this is going to be our tool for getting a yes/no answer of, does our candidate monoclonal antibody see the whole virus? So here you're seeing an overlay of one particular field. So here's on the green channel, here's the virus infected with GFP. And we've also taken those same cells and we've done some steps I'm skipping, fixing and then permeabilization with a mild detergent so that antibodies can get into the cell, but we don't want to really fry the cells or denature proteins or change proteins too much. So a little bit delicate. And now we're staining with, on the red color with a second antibody, we're staining to see whether our candidate human monoclonal antibody binds to those cells or not. And then here's the overlay, and we've got another color in there, DAPI, which stains the nuclei of all cells. So you can see there's a lot of uninfected cells around all those blue ovals. And then there are these nests of infected cells, and those same cells are lighting up nicely with our monoclonal antibody. So we've answered the question that, is this IgG VZV specific? Is yes, we got a smiley face. So how are we going to figure out which protein it is? So same basic technology. This is a little cartoon of the VZV genome, and you can see these numbers that go up to about 70. ORF is open reading frame. So part of the backstory here is we've actually cloned all 70 of these proteins, of these genes, into a nice little vector using PCR and cloning. And actually, they're all fused to GFP. So we're still using GFP, but switching gears a little bit now. We've got each one of these single individual proteins cloned as a fusion of GFP and just sort of a routine workhorse vector. So looks the same, but it's a little bit different. So this is pretty much my last data slide here. Is what we've done here now is we've transfected cells, transient transfection with a cationic lipid reagent. Just mix some plasmid and some cationic lipid reagent, dump that onto some cells, and this has this protein number 9 from VZV. So VZV ORF9, and as I mentioned, it's a fusion protein with GFP, and you can see some cells are positive and they're lighting up, they're expressing GFP. We forced them to. And now here we're staining them with that same candidate monoclonal antibody on the red color, and we can see that the exact same cells that are expressing this one VZV protein ORF9 are also expressing, are also being lit up and have binding from the monoclonal antibody. So now we know which protein it is. This particular one doesn't happen to be on the surface, but we've actually got a ton of these, and some of these are surface proteins, and not going to take you through all of that. So just at the very end, just sort of how would we maybe improve or optimize an antibody once we have an antibody? I sort of showed this cartoon before for, neutralization, viruses binding on the outside of the, antibodies binding on the outside of the virus, or this opsonization where it's making it attractive to be sort of eaten by an eater cell. Other ways that antibodies can act is they can actually sort of cause the immune system to sort of attack a virus-infected cell. If this is a virus-infected cell here in the middle, it's getting ready to pop out thousands or tens of thousands of daughter virus. So if I can execute that cell, if I can kill that cell with using antibody as a bridge to various T cells or NK cells or macrophages, or even the complement system to come and attack that infected cell, may do a little short-term damage by killing that cell, but it's going to reduce the yield of daughter virus. And there are many ways that antibodies can be changed or improved. I'm not going to go into that here, but so-called bifunctional antibodies. So here the antibody combining region of one half has been replaced by an antibody combining region from another half. So you can sort of get maybe synergistic potency. And there's a lot of changes that people now are making to the back end, to the FC part, to either improve the half-life of the antibody. So a lot of the monoclonal antibody products that you'll see advertised on TV actually have these, improvements in them, or to perhaps decrease some undesirable interactions with other cells. This is the naked capitalism part. This is just, there's a lot of slides like this and a lot of enthusiasm, but you can see that fully human monoclonal antibodies or humanized monoclonal antibodies are projected to, be big business. If I start putting a mouse monoclonal antibody into somebody, like these pink ones, these murine ones, we actually recognize, our immune system recognizes the mouse antibodies as foreign. And we end up getting our own immune response, our own antibodies anti-mouse, and that's not good, and they lose effectiveness. So the idea is that with a fully human or humanized monoclonal antibody, we'll have less, longer durability. We'll be able to use the drug longer and not get antibodies, against it. And I think that is, well, summary. Okay. So for us, it's all about the specimen. When you're doing this human monoclonal antibody work, we're letting nature do the work for us by localizing the B cells in the rash. The COVID work was all done with hot blood samples from recovered patients. So people who had recovered, but recently, and they had a lot of SARS-CoV-2 specific B cells in their blood. This uses very mainstream wet and dry lab skills. So single cell sequencing, carefully follow the 100-page protocol, and man, it's expensive. In our lab, it's really mostly doctoral level people who are doing this hands-on, but in other labs, people with other degrees are handling this. The data processing is a lot of bioinformatics. And then the designing the candidate antibodies is in silico, more bioinformatics. And you can either make them yourself. This is pretty mainstream stuff, cloning, sequencing, transfection, and purification. But we find it easier to order by takeout ourselves to be cost effective for this step. Fluorescence microscopy, which implies skills in cell culture, routine plasmids and transfection, and safe use of live viruses, pretty mainstream stuff. And then the plasma tools for the fine specificity, again, pretty mainstream stuff. And sky's the limit with antibody engineering. So let me, I think that's the end, and happy to take any questions if there's any time.
Thanks, David. Well, I think looks like we have a question in the chat right here from Lori. Why don't you just go ahead and ask it, Lori?
Yeah, Dave, this was wonderful. Thank you so much. So, getting the patient to make the antibody, hooray, that's a proxy for it being effective. But I'm guessing some patients make not great antibodies. And other than doing the whole workup, is any comment on that?
Well, if you're immunocompetent, you eventually do kick shingles. Now, we don't know really why that is between T cells and antibodies, maybe other parts of the immune response. But you're right, the art of getting a therapeutic antibody is in screening through probably scores to hundreds to thousands of candidates. And sort of not shown here are things like dose response neutralization assays, and finding that winner and then maybe even tweaking it a little bit more. I think what you're sort of intuiting is maybe we should mine the repertoire of people with mild disease or asymptomatic disease. And there is some, I think, versus people who had a severe course. And there is some, I think, rationale for that.
Keith.
You, David. That was an awesome presentation. Really appreciated that. I did have two questions, but I'm just going to ask one and let other people take advantage of the time if it's available. In one of the last slides, you went right to open reading frame number 9, protein.
Right.
Did you test for all 70 and that was the best
Yeah.
result? Or
So imagine a 96 well plate. It's one of those black plates that's friendly for the fluorescence microscope. And each well has like ORF 1, ORF 2, ORF 3, ORF 4, ORF 5, et cetera, up to number 70. And we do a screen. So we do a lot of that in the lab. And that's how we got it.
Thank you.
Okay, we're going to do one more question. This will be Todd's, and then we'll get on to our next speaker. So Todd asked, well, Todd, why don't you ask?
Sure. Thinking entirely about our project. So these current antibodies that you have, they could form seeds, right? Those, if you get the structures for then de novo design, and then that's going to kind of pepper antibodies all over whatever antigen you fed it that you could then screen for blocking and these kinds of things.
Yeah, I think what you're sort of intuiting is can you do a mutational scan or mutational, either random or directed mutants in an antibody to make it hotter? And yes.
Not necessarily hotter, but there could be unexplored epitopes that you're unaware of that could be more effective blockers. So de novo design would
so I think Todd, what Todd's getting at is the competition now. So we got this repertoire of 10 to the 13th antibodies per person, but actually now in the days of AI and AlphaFold is we can actually design a blocking protein kind of from vapor, from nothing, that will bind to whatever epitope I care. Here's the envelope protein sticking out on the side of BZV. I can just ask I can't, but some software genius to design me a new protein that's going to bind that and block it. Yes, that is out there. I think that there's always the concern for anti-protein antibodies or an anti-protein immune response. So some Frankenstein blocker protein that David Baker's lab cooked up might be really tight and have high affinity, but it might not be as long lasting as a therapy because we might generate our own immune response against it.
Yeah.
Cool. Okay, well, hope you all have a great day. I'm going to sign off and have a successful hackathon and a learning session.
Thank you, David. We really appreciate this.
Okay, bye-bye.
And good luck with finding those bits of the rash.
Bye.
All right. Well, now I'm really happy to introduce our second speaker, Dean Lee, who I actually met because I have followed and read his posts on LinkedIn, and I advise all of you who are interested in computational biology or bioinformatics to look him up on LinkedIn and follow him because he has been providing really excellent career advice. And it's a great, anyway, it's not always you learn things from social media. So I really advise you to look him up. And Dean, I'm going to let you go ahead and introduce yourself more. And do you want to check your slides quick, or I guess probably just get started?
Yeah, we can get started right away. I will share my slides now. Thanks for the introduction and thanks for having me here. Let me know. I'm gonna also go to slideshow. All right, do you all see the slideshow?
Perfect.
Perfect. All right, so before I even get to the slides, I just want to tell you a little bit more about who I am, what I do. My name is Dean. I am right now a computational biologist working at a pharmaceutical company called Novartis, which I'm in the Cambridge, Massachusetts office here, but we have offices worldwide. And I work specifically in the oncology department, working with other scientists who are either at the bench or also data scientists like myself to basically create better drugs for cancer patients. I personally work with a lot of omics data, specifically RNA-seq and single-cell RNA-seq data. That's basically my bread and butter. My entire day revolves around that, and that's what I end up talking about a lot on social media as well. Just because Sandra had mentioned my LinkedIn activity, I basically write content on LinkedIn as a way to teach people and to reveal a little bit more about what it's like to be a computational biologist in industry. That's one of my goals in writing any kind of content on there, is just so people know what it's like to be here. Because when I was navigating to that path, there was just no information out there. I had to like dig really hard to find even one person to talk to. Okay, so what I want to talk to everyone about, this is a longer title, but it's basically how do you navigate? It's 2025. Things have changed a lot, even just from 5 years ago. How do you navigate to a computational biology role? What are the different pathways that you can take to get there? And what my thesis is that really you want to focus on three things, which is domain knowledge, data types or data modalities that you specialize in analyzing, and data analysis methods that you are comfortable analyzing those data sets with. So focusing on those three things. But before we even get there, there's a bit of a setup that I want to go through, because I don't think it's necessarily self-evident what computational biology is and why it's important. We did the intro. Let's talk about we're going to talk about the century of biology, which I'll describe what that is. We'll talk about roles in computational biology. When I draw on the screen, you all see this, right?
Yes.
Perfect. Okay, perfect. All right. Roles in computational biology, and then pathways in computational biology. How do you get to those roles? And then at the end, I want to give a concrete example of basically what is the ideal path at this moment, which is going to change from year to year. But right now, if I were to carve out a path into computational biology as a college student, what would I do? And this is with a lot of hindsight as reference. Okay, so all right, the century of biology. Let me just erase this. The century of biology is a phrase taken from a quote by Craig Venter and Daniel Cohen from a while ago. It's more than 20 years ago. And I won't read the whole thing, but basically it's saying that the 21st century, they were predicting this would be the century of biology, where biology really shines. And this is getting to the point of, we're understanding enough of biology where we can begin to almost engineer it. And once you push a field into a realm where you can begin to optimize and engineer it, then that the innovation in that field starts to take off. So while combustion, electricity, nuclear power define scientific advance in the 20th century, in the 21st century, it's going to be the biology of genome research that will define this century. And I don't think anyone really knew what they were talking about in 2004 when they said this. The Human Genome Project had been completed. People were super excited about what you can do with human DNA sequences. But really how that's panning out is that now there's an explosion of biological data, and especially of sequencing data, because sequencing has gotten so much cheaper year after year for the past 10, 20 years, that now there's too much data and too few people who are equipped to really handle that data, right? And so to me, this is what's exciting about being in the 21st century and the century of biology, is that now it's an open game where people who want to get in this game, they have a chance to do it. Okay, so the century of biology really needs computationally equipped biologists. I know I presented myself as a computational biologist. I'm talking about pathways to get into computational biology, but really I'm also speaking to just people who want to be biologists in general, because I don't think it's possible to do biology in this day and age without at least being a little bit computational, just a little bit of Python, a little bit of R, a little bit of knowing how to visualize your own data. I don't think that's really optional anymore. So biology is becoming increasingly more data intensive. All biologists need at least basic computational skills to handle this data. And I'm seeing this right now, every day when I look at job postings on LinkedIn to learn what's on the job market. More and more job postings for bench scientist roles also ask for basic computational roles. So you don't need to work officially, your title doesn't officially have to be a computational biologist to benefit from having these roles. All biologists really will benefit from this. Also, biology education, because I've been looking around at what people are learning in school these days. Biology education, especially at the college level in the US, has not really kept up with this need. So part of that is because a lot of the curriculum is still mostly designed to fulfill pre-med requirements, which then there is no computational requirement as part of that pre-med set of requirements, right? And so what happens is if you are studying biology and you want to be computationally equipped, then you have to figure out how to acquire these skills for the most part on your own. And that is with the help of a lot of free open resources already online. So it's not like there's nothing to help you to do this. All right. What are the different roles in computational biology? There are actually so many. I had a hard time getting to a list that I think people would agree on. There are many more titles that I could have put on here that I didn't put on here. These are just the 5 major flavors that I'm putting on here. I'd like to talk about bioinformatician and computational biologist as basically the same thing. Some people argue that they're different, but most people use those two terms interchangeably. You also have data scientist and machine learning scientists, which I think are kind of the same thing. And then lastly, you have data engineer. So now that we have these 3 groupings of the 5 titles, I want to talk about what each group really does. So if you're a bioinformatician or a computational biologist, you are taking data collected from some kind of biological tissue, right? Cells or tissue. You're looking at imaging data, you're looking at sequencing data, maybe it's DNA sequencing data, RNA sequencing data, TCR sequences, T cell receptor sequences. You're looking at maybe protein data, proteomics data, really any kind of data that is, this is how I define it, any kind of data that is too large to analyze in Excel and requires some kind of coding, some kind of programming to analyze it. That is the job of a bioinformatician and computational biologist. With their work, I would separate it into 2 main categories. One is what I call upstream pre-processing of the raw data files. And then the other part is the downstream data analysis to generate insights. So those are often, even in industry, when we're hiring for bioinformaticians or computational biologists, a lot of times we hire for those 2 roles separately. We think of them as 2 jobs that are large enough that each can be its own person. And the upstream pre-processing of raw data is exactly as it sounds. When you have data coming off of a imaging microscope or data coming off of a sequencer, it's not in a form that you can begin to analyze right away. So you need a bioinformatician, for example, to run those raw files through some data pre-processing pipelines to massage it and get it into a form that then they can hand off to another bioinformatician or computational biologist. So then they can begin to analyze that data to extract insights from it. Okay, so that's what the first 2 bullet points are doing. And then you have the data scientist and machine learning scientist. You'll see these job titles. And when I think machine learning, I'm thinking about methods like, and machine learning and data science, I'm thinking about methods like logistic regression and deep learning methods, right? And regression trees and whatnot. And there's a classic defined set of methods that you can take in a data science or machine learning class now in probably most universities in the country that give you these techniques that you can then apply to biological data. So I consider these to still be roles in computational biology, even though your training may not be in biology to begin with. Maybe you're coming from a statistics background, or you're coming from a computer science background. If you've picked up these nice data science and machine learning techniques, and you want to apply them to biological problems, to biological data, and to answer biological questions. So you have that team of people, that category of people who may end up doing it kind of the same work as the downstream analysis that I mentioned that bioinformaticians and computational biologists would do. And then in the last category, which is the fifth bullet point, you have the data engineers. And this, I think, is kind of its own thing. So because there's so much data now generated in the life sciences, it becomes very hard to manage very quickly. You easily begin to have terabytes of data that you need to move around very easily, very quickly, and very securely. You also need to be, you also need to be able to access that data in a cheap way, because if you're storing data on AWS in the cloud, and you're like pulling data and putting data in there all the time, you get charged per action that you put on there, right? And so you need a data engineer to come in and basically optimize this whole system with which you're storing data and accessing data in a efficient and affordable manner. So that is a lot of those people then are definitely not coming from a traditional biology background. They're coming from some other background, or not even a biology background, right? They're probably engineering or like computer science people who took an interest in the life science industry and want to become a data engineer for the life sciences. And all of these roles that I'm talking about are often overlapping. If you go look at job descriptions now in this field, you'll see that sometimes people just blend all these terms together. You might even see jobs that are basically cover all 5 bullet points that you see here. They basically want someone to do everything that I talked about just now. So these are what I think are the major flavors of jobs that you can get in computational biology. There are, like I said before, there are just too many potential job titles that I could have gone over. And so I wanted to give people here a tip of basically how I study the variety of roles that are available. The best way to understand what are all the roles in computational biology out there is to study the current job postings. And I do this at least once a week, where I go on LinkedIn, I click on the jobs tab, I go to the search bar, and I put in these 2 bullet points in there. These are Boolean search terms where you use and or logic. And feel free to take a picture here or write it down for yourself. Everyone can do this. You just put these search terms in there. And this has been what I've been using to pull out any job out there that is potentially a computational biology job. And I skim read them pretty frequently to just get an idea of what are people hiring for now, how are these jobs transforming over time. And I set up daily notifications for these search terms so that I just get these job postings delivered to my inbox, basically. And I'll say this is, I think, maybe one of the most powerful tips I can give people, because let's say you start college now, in the fall this year, and maybe your university offers a 4-year computational biology degree, and it's really well taught, and it's been a program that's existed for many years. The job market now that helped you decide that you want to do computational biology is going to be very different from the job market that you graduate into. And the only way that you're going to know how to navigate your time in college to be best prepared for these jobs is if you keep watch over the LinkedIn job posts over the course of these 4 years. So then you build up this internal intuition for what people are looking for at any given time. All right. So what are the pathways in computational biology? I think of them as 3 major categories. There's industry, academia, and other paths that are not life sciences. So in industry, there's 2 major buckets of jobs that you can get. You can get jobs in a big pharma, or you can get it in a small biotech. And there's some contrast between what it's like to work in these 2 environments. In pharma, you typically have better pay. It's a much more stable environment. Things move slowly because there's a lot of red tape. There's no publishing requirement. The scope of work is very defined. you tend to have larger teams where everyone's role is clearly defined and people try really hard to not overstep their role. And then you have the smaller biotechs. These are companies that have been around for just a few years. They may have raised a couple $1 million or tens of millions of dollars just in the past 1 to 8 years. And they tend to have employees in the range of, 3 people up to maybe 100, up to maybe 200. They're quite small companies. These companies value speed over everything else. They want to see practical skills more so than credentials. They've moved very quickly. People in these companies get to wear many hats. So in a few slides ago, when I had the different roles in computational biology, it's very possible that you have one person in a small biotech who's wearing all 5 hats. And then biotechs are also inherently just less stable, because if you run out of funding and you don't get more funding, then you have to lay off people, right? So there's more frequent layoffs. It's a little bit more unstable. And then there are some other roles that you can go into in academia as well. If you take the PhD path, you might end up doing a postdoc in computational biology. And afterwards, you might decide that you really enjoy the academic environment and you want to continue to work and there are roles for computational biologists who are staff scientists, who are just regular employees of the university working in labs. I think that might be one of the least talked about roles, but one of the most satisfying roles, because it's a 9 to 5 job for the most part, and you still have your hands on the most exciting data that's coming out in the world, right? Because you're in a cutting edge academic lab. And then of course you have your professorship route. Not a whole lot of openings, but if you follow through with the PhD postdoc route all the way through and you land a professorship, that's another path that you might have. A lot of people that I know who originally aimed for computational biology, sometimes they are poached by other industries. And this is what I mean when I'm talking about the additional paths. So you have, what happens is, because as a computational biologist, you do have to know statistics, right? And data science and machine learning. And all of those skills translate well into quantitative roles outside of the life sciences, right? So some of these people, they just go into consulting. They go and work for McKinsey and BCG. They go and work for Google and Amazon. Google has a life science division called Verily Life Sciences. A lot of people get poached in their branch. So these are the 3 major categories of potential pathways that I think computational biologists can go into. Now I want to get to the meat of my presentation today, which is how to navigate computational biology pathways. Like I mentioned in the title, there's 3 things that we really should be watching out for. One is domain knowledge, and that is basically the niche in biology that you occupy. Are you a specialist in cancer biology, or immunology, or neuroscience, or evolutionary biology? Domain knowledge will not go out of style, even at the age of ChatGPT, because ChatGPT doesn't know what hasn't been discovered yet. It doesn't know what hasn't been published yet. And for you to work in the frontiers of your niche is for you to be positioned to have the domain knowledge that you will need to stay in demand. So pick something that relates to human health. I think that's probably the most marketable to specialize in for your domain knowledge. And then the second thing is data modalities. This is your data stack, right? Almost like when you have your business card with people and you're a computational biology, what are the data types that you can analyze right now today? And the hottest data modalities right now on the market, I listed them here. It's RNA-seq, single-cell RNA-seq, spatial proteomics, and imaging data analyzed together. So as many of these that you can analyze in an integrated fashion, the better. Then number 3 is data analysis methods. With every data modality, there's usually a set of established methods that you don't have to invent from scratch. They are often available as packages. You do need to learn how to use them for each of the data modalities that I listed above. And of course, you want to learn Python, R, and command line. You don't need to do Python and R at the same time. You just start with one and then eventually pick up the other. That's totally fine. But from what I've seen, you basically eventually you want to know both Python and R. And then statistics will never go out of style. You're always doing it. You're always testing hypotheses. You need to have a good sense for how the numbers are distributed in your analysis and whether that p-value even makes sense to you, for example. And the machine learning topics that I had mentioned before, that you can just Google. I mean, there's all these free resources online now to cover all of the statistics and machine learning topics that you would need. And as a bonus skill that you would want to have to navigate computational biology pathways effectively is communication, telling a story. Just because you are incredibly accomplished as a scientist and you've solved some really hard problems, if you don't know how to present it in a short amount of time very clearly to people who have money that you might need or opportunities that you might want, then they're basically going to assume that you didn't do anything. And so being able to present what you've done is maybe the most valuable skill on this slide. Storytelling, communication. And then, of course, there's a lot of project management because computational biologists never really work alone. You're always on projects involving a few or many other people. And things can get very hairy and complicated if you're not actively managing how each part of the project is getting done. And lastly, I'll just say that degrees and credentials alone are not enough. They certainly can be helpful sometimes. They certainly will never hurt you, but don't go into getting degrees or credentials thinking that that alone is going to get you a job in computational biology. It's not going to work. All right, I'm almost done here, and I hope that we still have some time for questions. So the main point that I want to drive home today is that because computational biology is so quickly changing and interdisciplinary, traditional higher ed training doesn't have the speed to really adapt to how quickly the whole field is changing. It's great for providing the fundamentals, the statistics, the biology, the machine learning, but you still need to do a little bit more on top of getting your higher ed training to walk one of these computational biology paths. I also say that there are many nonlinear paths. There is no one path forward. There are people who have been at the bench as a biologist forever. They're doing their second postdoc and they're asking me, is it too late for me to go into computational biology? It's definitely not, because you already have the biology piece, which I think is actually the hardest piece, and you can pick up the other pieces in time, right? And you can always transition. I mentioned before, focus more on skills than on credentials and prestige, because we can never hire people just because they went to Harvard or MIT and they went to a famous lab. That never happens in this field. I know that does happen in other fields, but not in this field, because we still need to see that you have the skills that's actually going to allow you to succeed on the job. There's no replacement for the hard technical skills. And lastly, your growth in navigating these paths comes not just from having the knowledge and skills, but also from communicating your work, making your work visible and engaging with the community. And this is the path that I would take now if I were to go back in time as a college student. I would go to college, I would major in statistics and minor in biology. And to get the most important biology experience, I would work in a biology lab all 4 years and apply my statistics skills to help the people in the lab analyze their data. Then after college, I would get a research assistant job. I would use the tuition reimbursement to get a master's for free, because it's just good to not take on too much debt. Some of these master's programs are so expensive out there. Don't go into debt for them, please. You can work and do a master's at the same time. The one that I would do is the one at Georgia Tech, which is a master's of science and computer science, because if I studied statistics in college, then I have this computer science gap, and I can fill that in with a master's from a reputable school. This particular program has been run very well for many years, and it's total $7,500 for the entire degree. And so I think people don't promote this program enough. And then afterwards, if I still want to get a PhD, I can always get a PhD, and I'm actually more competitive now for a PhD because I've done more years of research and I have a master's in computer science. Or if I find an industry job at that point, I would just jump straight into industry. So I think we're out of time. I only have one more slide that I don't have to talk about. I will stop sharing now, and we can take some questions.
Thanks, Dean. I can tell you've studied this a lot. It's great because some of the things you highlighted as technical skills and knowledge and data modalities were just in the previous talk. So it's really
Good.
spot on.
Yeah.
So we have 2 questions here, I see. Keith, why don't you go ahead?
David, and coming from the other end of the spectrum, having been through industry for many years and been in the defense industry, I wanted to ask you to perhaps comment on data security and computer security, if that's becoming more of a concern in your industry.
It's not. Like the word security doesn't come up that much in my line of work. I think when we think of security, we're mainly thinking about when we run clinical trials and we're collecting data from patients, then that information is like super guarded for privacy reasons. So like no one can really touch it except for a few people in the company. Even I can't touch it just because I'm in the company. So that's what I think of when I hear the word security. Now, I imagine you're talking about like national security, which is a niche that is growing. I know there's I think earlier this year there was like a bill that was proposed that basically focuses on biosecurity as a national security like initiative, right? I don't know where that bill went, if it's making its way through Congress, but I think that's actually a pretty outstanding report that it seems like dozens or hundreds of scientists worked on to put together to propose that biosecurity needs to be a priority. That's really all I know.
I can answer that a little bit too. For that biosecurity bill, some of that information is going through some of the current appropriations process. And their
Nice.
view is that biosecurity isn't just computer security like we think about it, it's also biotechnology and funding biotechnology. Just a few weeks ago, many of us who are principal investigators on National Science Foundation grants got a notice that we are now required to take a computer security course, 4 modules that are like an hour
Oh, wow.
each. Yeah, and we have to do this by October, and all the people who are applying for new grants will have to do this by this October as well. So we have another question here from Afshin.
Oh, hi. Yeah, so uh, you told us to major in statistics and minor in biology. Is it because of the skills that we're going to learn, or is it because of the hiring managers, they want this or something?
It's both. The hiring manager wants the skills. And okay, so back in the day, a couple of years ago, I was trying to decide because I hadn't really taken any stats in college, and I eventually got a second master's in statistics. And I was debating between like data science or statistics for a master's. And I basically saw that data science was a lot of it just felt fluffier, like people were using a lot of trendy words. And I wasn't sure if the students were actually learning anything. Whereas statistics, it was just like it's grounded in theory. You actually just do the math and you can see the results, right? And that is something that I still use every day. So that was maybe 5 or 6 years ago that I got the statistics training. I still do that. I still use statistics today in my work, whereas I don't really use as much like data science, the flashier stuff at work. So, which is why, and this is the slide that I showed you isn't telling you what you should do. That's just an example of what I would do if I were 18 right now and starting college. I would do statistics and biology.
Do you think, okay, I was thinking about taking my major as statistics too, but do you think I should minor in biology or just something maths related that has a quantitative bioinformatics?
Yeah, I would say also applied math would work just fine as well. You just want to have like you want to do your linear algebra in college. Like the hard stuff that you need to sit down and focus for a long time and to learn, you want to get that done in college because then you can always learn the easier stuff later on. So if you wanted to do like statistics and then maybe it's a very math focused, math heavy statistics major curriculum, that would be great. Or you want to do statistics and a computer science minor, but then also just make sure that you're taking biology electives and working in a biology lab to get your biology piece. That would also work. You can be flexible with how many pieces you're trying to pick up in college. And I think it's actually quite hard to have all of those elements built into 4 years of college, which is why in my path, I designed it so that I also have my master's degree to kind of fill in more gaps along the way.
Thomas, you, sorry.
Yeah, I just want to follow up on that. For community college students, I think you have a unique opportunity because this path is taking an approach for a 4-year college student. But you can go and get your associates in biology or statistics and then go to the 4-year college and major in statistics or biology because it's only a 2-year degree, not a 4-year. So if you major in biology or a STEM field for an associate's degree, then you can go switch to statistics or something else like Dean is talking about. So I just wanted to point that out, that it gives you community college, I think, then an advantage in that you can get your one associate's degree in one area and then pursue the other area at the 4-year degree.
Thanks, Thomas. Dean, I don't want to keep you too long, but we have one person thanking you for pointing out that if you're working at a university, you can often use tuition reimbursement to pay for your courses. Many of us have done that. I took statistics that way in PChem. We also have a person asking you if you can take one more question about what you would advise a biochem graduate with limited industry experience.
It's hard to answer that question without knowing a lot more about what your exact individual situation is. But I think what's always good to do is to take an inventory of your skills. So you have you study the current job postings and you can take those bullet points in those job postings to know what people are hiring for. And then you have also a list of your skills and you just compare them to see what's lacking. And the bullet points that you're lacking, then those are the ones that you can potentially take a few classes for. And then once you have, or you can join. I think it's always a good idea to do a few years as a research assistant in a lab. It's just a very rich scientific environment. And you can do that for 1 or 2 years to shore up more of those gaps, and then you can begin to apply for industry jobs.
Thank you. Well, everybody, let's thank Dean Lee for sharing his expertise in navigating the computational biology pathways. I really appreciate you helping us out and coming to talk today. And we're going to have a short break now until the 1/2 past. And at 1/2 past, we're going to, every group is going to give their presentations. So I'm going to be opening up the breakout rooms and giving you time to have a short break. And thank you once again, Dean. I really appreciate it.
Yeah, thanks everyone. Thanks for having me. All right.